cells should be in the highest amount. White blood cells, if they exceed normal limits, something's wrong. All right? So if you get to look at a blood smear that has been stained, and depending on whether you're looking at someone who's sick or not, will determine how many of the white blood cells you see and which of the white blood cells you'll see. So all of these elements formed, keeping their properties. Okay? I have my red blood cells. These are considered biconcave. See how it looks like they're trying to show it dipping in the middle? Okay? Almost looks like a donut. All right? This is considered biconcave. So I've got my red blood cells. I've got my platelets, which are not a complete cell. They are simply a fragment. Then I have my leukocytes, the white blood cells. They are classed, the categories that they exist in. They can either be a granulocyte or an agranulocyte. Granulo. What does it kind of bring to mind?
Sand.
Sand. Granules. All the white blood cells have granules. It's just that when we first began to have the ability to look at them, when scopes were not as good as they are now, all right, it looked like some of them had the granules and some did not. So that's why they got termed the granulocyte or an agranulocyte. There's a total of 5 altogether. The ones that are listed as granules, neutrophils, eosinophils, basophils. Agranules, lymphocytes, monocytes. Person who is trained in looking at blood smears, they would be able to take a sample that's been given, create the slide, stain it, put it under the scope, which would be a special scope that they have, because they're trying to determine a count, a number. They would want to try to determine for that patient what is the number of red blood cells that they have, what is the number of platelets they have, and then which white blood cells are present and in what amount. Does that make sense? The reason for trying to determine a number, if the numbers do not fall in what is determined as our normal range, something's wrong. That means there could be something wrong with the red blood cell count. There can be something wrong with the white blood cell count. There can be something wrong with the platelet count. And that results in physical problems. In other words, it's clinically significant. All right? Now, this is trying to show you a representation of what these leukocytes look like. So, neutrophils, eosinophils, right here, basophils, and then they're trying to show me lymphocytes and monocytes. They're showing you that for the leukocytes, they maintain their nucleus. They keep their organelles. Red blood cells do not. Leukocytes keep their organelles. Red blood cells do not. Platelets don't have them to begin with. So how is it that we actually get to take a peek at someone's blood? Anybody in here ever had blood drawn? Anybody in here never had blood drawn? Never? Even the little stick on the finger?
Oh, I've had that.
Okay.
Yeah. Never had
Good luck. I don't like it. Okay, I really don't. But I have to sit there and look at it. Okay? Whichever one they're choosing to go into, it's like a train wreck. It's like you can't help but watch it.
Nope.
Okay? And so, you know, I don't like it, but I'm like,
I hate needles. I turn away and I close my eyes.
I don't like them either.
This is me the whole
I
time.
really don't. And so, here's what they'll do. Like, those of you who've had to have blood taken, there's usually, like, you know, this tube, depending on what they want to do. There could be multiple tubes. And you'll see that it goes into the tube, and usually there's a little substance at the bottom, and that's for preserving until it gets to the lab, okay? And you'll see them take it and just do a little shake with it, because they're trying to preserve the material. Then hopefully they label it correctly, all right? And it goes to the lab for the testing. In the lab, there's this really cool machine. Anybody ever seen a centrifuge? All right, they're really cool. Don't ever open one until it completely stops. Okay, before, let's see, I guess it was right after I had earned my master's degree, I went to work at a biological lab. Learned that. Didn't happen to me because I kind of knew it. But I saw some other people who would try to open that centrifuge too early. That's not a pretty sight. And I would not want to have all those blood samples all over me. Just saying. Okay? Now, they take the tubes and they position them into the centrifuge, and it begins to spin. And when I say spin, it's a very high rate that it spins at, almost like it would take off and go into space. And then it begins to slow down, and slow and slow and slow. The goal is to separate that material. For our blood, it separates based upon the amount, weight, that sort of thing, because the heaviest in number and so forth separates out first to the bottom. So when they're done, this is usually what it looks like in the tube. Those red blood cells, my erythrocytes, my RBCs, okay, they should make up somewhere about 45% of what's termed whole blood, all of it together. So that amount should be about 45%. This little middle part right here, this, what they call the buffy coat, because that's exactly what it looks like. Almost looks spongy. The buffy coat Those are my leukocytes and my platelets. They should only be making up less than 1% of the count. The remaining portion, which is the majority, is my matrix, the plasma. My plasma is carrying stuff such as nutrients, gases, okay? My red blood cells, they're pretty much their job, transport oxygen. We need a bunch of those. That's why there are so many. But what do you kind of notice about this buffy coat?
It sits in between the red blood cells and the plasma.
But it's only made up of my leukocytes and my platelets. Telling me numbers should be low. So if somebody were to have blood drawn and that buffy coat came out like this, something's wrong. And it becomes the job of now testing, staining, whatever the case might be, of that person studying the blood to determine what it is.
What would be wrong?
Well, it would depend on, all right, like right now, weather's kind of getting cooler, lots of mold in the air, lots of stuff flying around. A lot of us having our allergies flare up and that sort of thing. That means our body would be producing more of those leukocytes, such as eosinophils, and that would make that buffy coat bigger. And if they did a peek at that, like if they tested it and looked at it, and if it showed a huge number of those cells, then the doctor would be like, oh, okay, well, you know, your allergies have flared up. Let's increase your allergy med or whatever. Now, if it begins to show other things, like some of the other white blood cell counts, here's the cool thing about those white blood cells, where you've got neutrophils, monocytes, lymphocytes, neutrophils, eosinophils, all these different types. Each one is kind of geared towards specific invaders of our body. So they would actually have to test to see which one is high in number. Doctors could then say, well, let's check for blank and blank because this is what it's indicative of. Does that make sense?
Yeah.
So it's really cool how they use this information. When we look at our values, because I put this up here mainly because a lot of times when we have, say, the blood work done, and do any of you actually look at your blood work?
Nope.
Why?
Nope.
We don't know what we're looking at. You're going to know now because you're taking my class. And hopefully you'll be like me from this point forward. You don't let your doctor get to do anything without you knowing exactly what it is. For example, we may not know. We may look at it and be like, okay, well, my hemoglobin, blah, blah, blah. What is it supposed to be? What is the norm? All right. For example, pH. And if you've got your, let's say you've got your results that you're looking at, and you see that they have pH 7.8